All right, good morning, everyone. Welcome to today's, webinar on high dimensional tools. And we'll have an emphasis today on how to compare categorical variables or categorical parameters. And I will kind of walk you through a couple of slides just to kind of describe what we're going to do today. And then afterward, we're going to go into a live demo and I'll show you how we can create categorical variables and then use them to make comparisons between those variables in a high dimensional workflow. Okay, so my name is Jack Panopoulos and my email is right here, jack.flowjo@bd.com if you have any questions. I will say that during the session today, it's not moderated. I'm kind of by myself, but what you could do if you have a question is please use the chat box to ask a question. And usually what will happen is once you hit enter, the chat box will light up on my end and I'll be able to see that you've asked a question. I'll take a moment, stop, and then go ahead and I'll stop and answer the question and then move on from there. But just to give you an idea where the chat box is, I'm going to go ahead and type something into it right here. And then what will happen on your end is on your menu, you should see it kind of light up sort of in an orange color. And normally the chat box will be located underneath the more menu, or it may appear as a chat menu item on its own. Zoom is always updating, so sometimes the placement of these things kind of changes from time to time. So hopefully you see my message to you, hello everyone, and then you can use that box for for questions going forward. Today's session shouldn't be that long, but in any case, I would prefer that you ask questions via the chat box. Okay, so enough about the housekeeping. Let's move on to our kind of just a quick overview of the workflow, like what you would, how you would approach using these high dimensional tools in general. And then we'll talk about how you get to the categorical parameters and why that's important, okay? So in general, you want to clean up the data first. You don't want to have any dead cells, debris, doublets, things of that nature going in, because you're going to be using mathematical algorithms to identify populations of cells. And so they don't know the difference between dead cells or debris or doublets. So it's a wise idea to only feed clean data essentially into the algorithms so that what you get out in the end is something that is interpretable. You know that these are live cells. They have, components or principles about them that make biological sense. If you keep the dead cells in there, the debris, you're going to get some very fantastical results, and it'll send you down a rabbit hole that I'm afraid you probably don't want to go down. Okay, so clean up the data. Compensation also kind of included here. You want to be wary of your compensation in the sense that you want to make sure that your compensation is good for these larger panels. If you have a population or two that are relatively wonky, so long as it's consistently wonky, it should be okay. But you don't want to have a situation where, let's say on Monday, the data look fine, and then on Friday, you've got, I don't know, a plot or two where the populations are definitely skewed one direction or the other, because the end result is the algorithms are going to see that skewed population as being different, when in reality, it's the same. So you're going to have to do some backgating, extra backgating, so to speak, to figure out that, hey, these populations that Tisney or any of the clustering tools are finding is actually the same population that I was looking at on Monday. It's just that it's skewed. So just be aware of that. The other thing I should mention too, sort of in the cleanup phase of things along the lines of compensation, et cetera, is the transformations of your axes. So what I mean by that is the scaling, right? You want to make sure that when you use these tools, before you actually run them, you want to make sure that your parameters are scaled appropriately. You don't want to have a lot of white space on the right-hand side. You want those cells that are positive to kind of move as far to the right as possible without being all bunched up on the far right axis. Okay, same thing with top, up and down. You just want the data to be spread as good as possible to maximize the distance between negative populations and positive populations. And then that will result in better resolution of your populations in both, dimension reduction techniques like t-SNE or UMAP, and clustering algorithms like phenograph or XShift or FlowSum. Okay, so those three principles, I should say, are probably part of the cleanup phase. And then after you clean the data, this is where today's seminar is going to kind of focus on, or at least put an emphasis on this particular step here, adding keyword metadata. So you have to think about your experiment, think about all the experimental variables that you're testing. Some of them are obvious, right? You have disease versus healthy, or you have treated and non-treated, right? That's easy, that is obvious. But you may have other experimental variables that are present that you may not want to test up front, but are present, and it would be a good idea to carry those along. So for example, you might be working with, I don't know, 2 different cohorts of mice, right? And you have both male and female mice. Now, normally, let's say, you expect that the biological outcome for males and females is going to be the same. But regardless, you do have males and females. And so you might want to incorporate the fact that you have male and female mice into your categorical variables, even though you don't expect it necessarily to be something that would pop out, or that is something that you're immediately testing. Okay, same thing with like age of the mice, right? You might have young mice, you might have juvenile mice, and then you'll have like adult mice or elderly mice, right? So you may want to add that information as well, even though it may not be the primary thing that you're testing. Okay, so addition of metadata, super important. I put a step in here for downsampling. Downsampling simply means to reduce the numbers of events that you feed into the algorithms to some lower arbitrary number. In general, this is a wise idea because it accomplishes 2 things. Number one, it normalizes the numbers of events, right, across all samples. So when you want to make frequency comparisons between 2 tissues or 2 cohorts, it becomes very easy. If there's ratiometric measurements you want to make, it makes it very easy because everything's already, normalized that way. The other advantage that it provides is the fact that it speeds up, the processing of these algorithms. So t-SNE and UMAP are historically fairly slow algorithms in relation to other, algorithms that process your data. I mean, a lot of improvements have been made to t-SNE to speed it up, but it's still relatively slow compared to, say, clustering algorithms. So lowering the numbers of events that you feed into it is going to make it run faster. So it has that advantage. Now, there is a caveat there. Be completely transparent. Depending on how far down you downsample, right, how many cells you reduce, you can clip out rare events. So you kind of want to be careful about just how far you downsample, especially if you are chasing rare populations of cells, right? So don't downsample too far if you are going to pursue rare cells. Okay, and now obviously that's going to be a function of Poisson statistics to figure out exactly how many cells you need and whether or not the downsampling will cut them out or not. Okay, so after downsampling, we will go ahead and merge the files that we wish to compare together. This step is a concatenation step. So we basically take all the files, right? Everything that we are intending to make a comparison, either in t-SNE space or when we cluster the data, we want to make sure that we're doing it on one file, okay? And then that one file will get, t-SNE run on it or some clustering algorithm on it. And then we'll be able to tease apart which samples are which in that concatenated file. Okay, so the original presence of the files, or the original files that make up the merged files, the identities of them are not lost. It's retained, but you have to know how to pull out that information. And so I'm going to go into that as well today. Okay, so from there, after we do some dimension reduction on the concatenated file, TISNI or UMAP, maybe even TriMap, then what we'll do is we'll do some clustering. This, again, clustering is different from dimension reduction in the sense that dimension reduction gives you this two-dimensional image, right, of like a series of islands or blobs on a, on a two-dimensional background. Whereas clustering, you just feed the data to the clustering algorithm like Phenograph or FlowSum, and then what it does is it spits out populations for you in the workspace. So TISNI gives you a picture and clustering algorithms give you populations. That's the primary difference between those two techniques. Both of them are mathematical, algorithmic. But after you cluster, you may want to use the Cluster Explorer to figure out the phenotypes of those clusters, because oftentimes the clustering algorithm doesn't tell you what the phenotypes are. It just says this is population 1, 2, 3, 4, 5, blah, 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 and you don't really know what they are unless you do some digging. And the easiest way to dig through those is to use this plugin called the Cluster Explorer. So I'll get into this as well. And there's a module here within the heat map, actually, that will allow you to make comparisons, again, between your categorical variables. So if you have a disease group and a healthy group, and you want to see how the different populations align themselves, right? Are they more skewed toward the disease condition, or are they more skewed towards the healthy or the non-treated condition? The heat map down here, when you modify it, will allow you to figure that out. And so I think it's a very powerful tool to be able to explore differences between your categorical or your experimental variables. Okay, after we do that, we're obviously going to make some comparisons in dimension reduced space, and then look for differentially expressed population. So we'll actually, the way I have it on this slide is slightly out of the order that I will do it in, but I don't think that's super important. The general idea is what we're going to apply, and then of course how we're going to pursue those differences. Okay, so little emphasis here on the keywords, right, as categorical parameters. I kind of already went over this in a lot of ways, but think about how are we going to segregate different tissues, time points, or treatments that we have in a series of data files. You guys probably have very simple experiments, but you probably also have a lot of complex experiments, right? And you want to compile all that information, and you want to be able to make cross comparisons between each of the variables, right? I have a 6-month time point that I want to compare in the liver tissue, as well as the lung tissue, as well as the spleen, right? And then I want to look at that maybe in the context of a 12-month time point, right? Same thing, looking at those 3 different tissues. And then I want to look across that, let's say across males and females, or old mice and young mice, right? The permutations are seemingly endless. So we want to be wary of these things, right? The different variables that we have. And the way that we're going to track all of this information in FlowJo anyway is to add a keyword. Okay, 1 keyword essentially for every categorical parameter, or for every experimental variable, you should add a keyword. Okay, so if you're going to take into account the different cohorts of mice, right? You have, let's say, BALB-C and Black 6 mice. That is a keyword, right? The mouse cohort, for example. If you have healthy and disease, right, you could say, well, this is the condition, right, the disease state. So that would, the keyword would be disease state, and then you would assign different values to each state. Okay, same thing with a treatment, right? You treat cells with drugs or mice with drugs, whatever. The drug treatment would be the keyword, and the different types of drug treatments would be values that you assign to that particular keyword. Okay, add a keyword, yeah, for each category or experimental variable. That goes without saying. And then I highly recommend here that you do not use like text letters, like ABC. Do not use alphabetical symbols. Use non-zero integers. So like 1, 2, 3, 4, 5, right? Make it really easy, and then it'll help you in the long run. All right, so let's just see really quickly how you do this, or at least where you would encounter it. you basically would right-click on a file. In this case, you're going to right-click. We'll see how we do it in FlowJo here. I'll give you the demo of it, but you'll right-click on a file, and then you'll say Add Keyword. There's going to be a little menu that gives you that option, and then that will prompt you for a name. And then you type in the keyword name, and that will show you a new column in the workspace. So you can see here that my colleague has added 3 different columns here, all indicated kind of by this asterisk. So you have the order, you have something called a time point, and something he's labeled as tissue. And as you can see underneath each column here, the values are sort of just integers that sort of increase, right, one by one. So he has a time point, 1, 2, 3, 4, blah, 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 right? Different types of tissues. And then of course, there's a sort order that he is assigning to them as well. All right, so enough about that. Let's just go ahead and see how all of this works in FlowJo, and then how we can use it to our advantage when we're trying to use these high dimensional tools. Okay, so I'm going to exit out of the slideshow, and then we're going to come right here into FlowJo. Now, the example that I'm going to give you guys today is fairly simple. Okay? I didn't want to get it too complicated. Don't want to throw you for anything. I think it's a very elegant data set for this purpose. Okay, and I'm going to just collapse everything here really quickly. I recommend that you collapse your data files if you have a hierarchy underneath. So all of these data, for example, they've been compensated. They have a bunch of cleanup gates, and then there's a bunch of phenotypic and functional markers associated with different subsets of cells. So the gating's already been done, but collapse your hierarchy so that you can see all the samples here very easily. And then that just makes adding the keywords a quick job. Okay, so we're going to go ahead and right click here on a sample, and we're just going to say add keyword. Okay, and then from here, we're going to add some keywords that describe the experimental variables that we want to chase down in this experiment. So in my case, I have 2 files here that have not been stimulated, kind of indicated by the file name itself, right? No stim. And then I have another set of files here that have been stimulated with something called a toll-like receptor agonist, so the TLR stim. Okay, so very similar to LPS. Generally, when you dump this onto antigen-presenting cells, they will get angry and produce a lot of cytokine, etc. So we want to track those 2 variables, and then the other variables that are present here is the patient, right? Where the information comes in the form of the different patients. So I have a PID1, patient ID 1, right, and a patient ID 2. So I'm going to add that as a categorical or experimental variable here as well in the form of a keyword. So let's start with the stim first. Okay, so I'll just call this something like the stim condition, right, or stim type, something along those lines. Click enter. You'll get a new column here. And then we need to create another keyword, right, for the patient ID information. So let's right click again and say add keyword. And for this one, we're going to say, I don't know, I'll call it maybe the patient number. Sometimes the data files will have, a patient ID already, and I don't want to overwrite an existing keyword, right? So if patient ID already exists, I'm going to go ahead and just, swerve around it here by calling it patient number so that I don't have any kind of conflict that way. Okay, and in this case now we can say, all right, I've got my keywords that I want to use to track the differences between these files. But now it comes to a matter of assigning the actual integer values. Okay, so for the non-stimulated condition, what I'm going to do is I'm going to go ahead and add the value 1, right, to each. And then for the toll-like receptor files, the ones that have been stimulated, we're going to go ahead and add the value 2. Now imagine for a second that I had, let's say, I don't know, 10 files here, but had different sorts of agonists that were tossed onto them. So maybe I had LPS, right, or TNF or IL-1. Those would you know, just inherit different integer values. So if I had LPS treatments, I would just add the number 3 for those that were treated with LPS. If I had TNF treated ones, I would add the number 4, right? So on and so forth, okay? Because they still are talking about the stimulation condition. It's just a different type of stimulation that was added. Now, for the patient number, this is pretty straightforward. We can see that the first sample comes from patient number 1. The second sample comes from patient number 2. The third sample comes from patient number 1, right? And the fourth sample comes from patient number 2. So we sort of have alternating keywords here, or alternating keyword values, right? Whereas we have sequential ones over here. All right, so this step, right, on your non-concatenated, your initial files is extremely important. Any other variables that you can think of that exist in your data set, I would highly recommend that you add them to the data, okay? So now it's time to concatenate these files together. We need to merge them together if we want to run tools like Tisney or any of the other sort of clustering tools that we have available to us in the plugin drop-down menu. Okay? In order to do that, we could select all of these files sort of, you know, at the level that they're at right here and just simply concatenate them together. But I would advise against this, again, because we don't want to throw data that have dead cells, debris, you know, doublets, et cetera. We don't want that information in the concatenated file. It's better that we concatenate at some level in the hierarchy that's already been cleaned up. So I will go down the list here, right? I have a time gate. Okay, that's good. I get rid of the anomalies, and I have a singlets, I have a size, et cetera, right? I finally get down into my lineages. And it's really up to you where you want to begin the analysis. Obviously, the higher the level in the hierarchy, the more complicated or the more interesting, I would say, the results normally are. But you can focus on any compartment that you're interested in. In our case here, we have a 13 color experiment. So I'm going to, you know, it's not too complicated, but I'm going to keep it here at the antigen presenting cell level. And then we'll try to find a lot of, you know, the populations that we see here using Tisney or the clustering algorithms that we employ. Okay, now, as I mentioned, it's important to transform the axes. So I'll give you an example of one here that we ought to transform right off the bat. And this is CD1C, for example, right? This is a marker that marks dendritic cells, but it's not a very strong, you know, on-off type of a marker. As you can see here, right, it's a bit fuzzy what we're considering positive. Right? And this is what we're saying is negative. Again, the distance, the more distance you put between the positive and negative, the more you stretch this out, the better the results are going to be when you cluster this or when you use Tisney. It'll make the algorithm, you know, it'll help the algorithm to see what is positive versus what is negative. So I'm going to go ahead and just do a little transformation here. I'm going to customize the axis, and I'm going to try and get rid of that white space, you know, so that we really emphasize this stretching of the data, right? I don't want things to pile up on this axis. Same thing on the left-hand side here. Maybe I can kind of stretch this out a little bit towards the left so that, you know, we have some events, again, not pressed up too much over here, but almost touching. How's that? Right, something like this, so that we get better, you know, better separation between these guys. The bigger the distance, the better result. Now let's do the same thing. What do we have over here? This is per CP, Psi 5.5. This is CD123. So let's play around with this one as well. Just go ahead and kind of, you know, stretch this out a bit so that we have some a little bit more spread in the data. Okay, and then really what I want to do here is I want to pull this guy far to the left as possible. get as much space as I can between the two. So I could take some, I would probably take some more time to do a better job of this, but you get the idea. Okay, I'm just trying to expand the distance here a bit. All right, so you've made your transformations, and now it's time to pick the compartment. In my case, as I mentioned, we're going to pick the DR compartment. Now we want to concatenate the DR compartment not only from the non-stimulated file that we're working with, but we want to do the same thing with all of the other files that we have here at the same level, right? I want DR from sample number 2, sample number 3, and sample number 4. Okay, so the easiest way to do this is to highlight the DR compartment and then right-click on it and say select equivalent nodes. This is going to go ahead and highlight all the DR compartments, right, across all samples in that group. And then all you have to do after that is another right click where you say export concatenate populations. Okay, so we go ahead and click on that. It brings up our concatenation window. We want to make sure that we're on concatenate or merge rather than export, right? Because we want to merge the files together. Make sure you know the destination, right? Where the file is going, so that in case, I don't know, if you lose the workspace or whatever, at least you know where the concatenated file goes. Pretty straightforward. Now for downsampling, I did mention that you could downsample. You can do it using a downsample plugin, or you can do it directly within the concatenation window. And I prefer to do it in the concatenation window simply because it's faster. So this box where it says include events will allow you to downsample to whatever number of events you want per file. So we say include no more than, and then you tell it how many events per file you want. So I'm going to say, okay, I want no more than 10,000, and it's referring to the actual level in the hierarchy here. So I'm telling it, please take no more than 10,000 for the DR compartment. Okay, so it's going to take anything that's included in the DR. Anything above that is excluded, right? So if it happened to fall into the dump channel or the live or the size or singlets, blah, 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 right? None of those cells are going to get incorporated there. It's everything DR or downstream. All right, for the parameters, normally what you guys are going to want to choose is the second option. Okay, this is what you normally are going to do, all compensated parameters, because you're going to have data that come from different days, and you're going to want to preserve the compensation matrices that come from the different days. Okay, in my case, all of these files were compensated on the same day. And what's going to happen is I'm going to concatenate, and that file is going to get dumped into this workspace, and it's going to automatically pick up the compensation matrix that's associated with these files. So I'm going to leave this as all uncompensated, okay? But this is the exception to the rule. You guys are probably going to want to choose the second option. Okay, and then let's call this concat, I don't know, advanced maybe, advanced seminar, something like that, okay? And then from here, the last thing that I'll kind of cover in the concatenation window is this one, our categorical parameters or the experimental variables. This is very important, right? This is where we add those keywords that we had initially created for each of the files. This is how we wrap them into the concatenated file. So we go over here to the button where it says choose, and then we scroll down to the bottom. Usually this is where your keywords will end up. And then we find our stim condition, right? And we also find something, our patient number keyword that we created. So go ahead and highlight, right, the keywords that you added, and then you click OK. When you do that, the window will give you this little red X, and it'll tell you how many keywords you have selected. So in my case, I have 2. All right, so I have a question here. And let me see, I thought rescaling the axes was purely visual. Is that wrong? Does it change the data point values? It does change the data point values when you feed it into t-SNE or when you feed it into the clustering algorithms. So the t-SNE and the clustering algorithms are not using the raw bin data underneath. They're using what we call the scale space. So what you see visually, when that gets fed into t-SNE, that's the scale that it uses. Okay, now in terms of deriving medians and things of that nature, statistics, right? The effect that you see in FlowJo, that those statistics that are derived, it is a visual change. You're not actually changing the values of the data, but for the so for the median or the geomean or whatever statistics you're trying to derive, none of that changes. It's going to be the same as what you derive on the raw data. The difference here is that the scale space matters when it comes to it's like as if the algorithms had eyes, right? Imagine, it's a lot easier to see two populations that are spread apart with a lot of space in between them versus two populations that are sort of crammed together. So I don't know mathematically how that translates, but I know that it does have an impact here. And you will notice that difference if you do not transform and you have a lot of compact populations. Your t-SNE and your clustering results are not going to be super satisfactory. Okay, or at least it's going to cause you to backgate a heck of a lot more than you would if you had transformed it. Okay, so we're going to go ahead and click concatenate here, get our concatenated file. You get a little prompt to go to the existing workspace. You could put it in a new one or both. It's up to you. I'm just going to dump it into the current one. And then you close out of these. It's kind of a strange, I don't know, it's kind of a strange order of operations. The concatenation window doesn't automatically close after it produces a file. So you kind of have to close it. Okay. So now we have a concatenated file. I have a group in my workspace that catches concatenated files, so it makes it really nice. There is my concat. You can see here that it is compensated, and so all of the parameters that you will see here are now all compensated parameters. They're not uncompensated. Okay, and again, that was kind of the exception to the rule with respect to the data files that I'm using, right? Everything's done the same day, same comp matrix. So it just gets applied automatically to this guy. All right, so I advise you at this stage, once you have your concatenated file in the workspace, I advise you to make your categorical gates right away. So there's a couple of ways to do this. One way is manually, right? So I'll show you that way first. So let's just choose, for example, the stim condition, right? You see now we have the keywords as categories in the drop-down menu, right? Normally in our raw files, right? If I come over here to the raw files that we used to generate the concatenated file, if we actually go here, there's no sample ID, there's no, or stim condition or patient ID, right? There's no sample ID either. Okay, the concatenated file, on the other hand, right, happens to have a sample ID as well as a patient number and a stim condition, right? These are categorical parameters that we added. Now, by default, FlowJo always incorporates a sample ID. So this corresponds to each of the files that you have in the gates group in this case, right? All 4 files that we had. The way it gets concatenated is the order that you have it here. So we would be looking basically at, the non-stimulated, the first 2 here are non-stimulateds, the last 2 are the TLR stimulateds. Okay, that's cool. It's probably simple enough when you have only 4 files to track these and to understand that. But when you have multiple files and you want to make quick comparisons between different compartments, it is best, again, to have your categorical keywords. So that's what we're actually going to use here. Okay, so I'm going to shift over here to our stim condition. I'm going to show you the manual route, and then I'll show you another route to automatically gate these populations. Okay, so convert to stim condition here. Then I would change the axis from a bi-x, which it kind of defaults to a linear axis. And then I would go to maybe make this into a histogram. Okay, and then what you can do here is you can just simply, you can go ahead and draw a gate, and you can call the first population, right, was non-stimulated. So these are the non-stim. And then you can create another gate for the second set here, and you can call these, I don't know, the TLR stem or the stem, whatever the case may be. Okay, check your work when you create these gates. So we got to go back to our concatenated file. Make sure, right, we have 2 gates here, 20,000 cells apiece, right? Because in total we have 40,000 events, right? 10,000 from each file. 2 files are non-stimulated, 2 files are stimulated. Both have 20,000 events apiece. That looks good. So that's the manual method. You can imagine if you had like, 50 of these, that'd be a little, I don't know, a little bit problematic to try and gate those one by one. So there is another tool here that you could use. There's actually a couple of tools you could use. I'll show you the one that is in the plugins drop-down menu. So you guys may not have the list of plugins here. I'll describe how to get these in a second, okay? But the one plugin that I'm going to show you right now is something called AutoGate Categorical. This is a really nice, simple tool for creating gates on, experimental variable keywords, basically, okay? So you choose the categorical parameter. So in our case, let's go down to our patient number, right? We have patient 1 and patient 2. You can give the population a name. So I'm going to call it, instead of pop, I'm going to call it PT for patient. Okay. And then if these categorical parameters have discrete values like 1, 2, 3, 4, it'll make it so much easier for the algorithm. In my case, I do. And then we just go ahead and click OK. And now it's going to go and find the patient number, right? And then it says, okay, here you go, patient 1, patient 2. Now, if I reference the workspace real quick, you can see that didn't do such a great job here. We've got 20,000 in one and 19,000 in another. So I'm going to go here and I'm just going to look at the parental population to see what went wrong. Okay, number 1, I use this. I didn't linearize the axis here. Okay, so made it a little bit hard for the algorithm to actually, find 1 and 2. So maybe we'll retry this, but after linearizing the axis. Okay, so I'm going to go ahead and delete this. And then we'll just go back to AutoGate Categorical. Whoops, almost made the same mistake twice. Let's go back here to patient number, linearize the axis first. Okay, and then I'm just going to try rerunning it. I see that it regenerated those gates, but I have a feeling it could do a better job. Okay, and if it doesn't, I'm taking you down a rabbit hole that maybe you don't want to go down, but I think it's a very nice tool to quickly gate the populations. Yeah, unfortunately, it didn't do, it's just not doing as good a job as I wanted it to do, but you can always come back here and you can, adjust these gates. Another way to sort of get around this is, I didn't mention it, but in the concatenation window, I have that box ticked where it says spread distribution. If you untick that box, everything here will appear as a single line, and it makes it, makes this algorithm work a heck of a lot better. But in any case, you can correct it, and there we go, okay? 20,000 a piece for each. All right, now I wanna, I'm not really satisfied with this naming schema, so let's just rename. I really only wanted this to be named PT1 and PT2, and I forgot that it always precedes it by category name. So let's just do this, right? PT1, PT2. Simpler, a little easier for me to find. Okay, now you have your categorical gates, right? And you can mix and match these guys any way you want, right? So if you wanted to recapitulate what we had in this gates group, right, to get all down to the original files, all we have to do is drag and drop these gates under the appropriate locations. Okay, so we have 2 stim conditions. We just drag those underneath, let's say patient 1, right? Do the same thing here. We can drag those gates under patient 2. Okay, we could have done the reverse as well. We could have taken the patients and put them underneath non-stim and stim, whichever direction you want to go. But the bottom line is we have now recapitulated those 4 files, right? We have the non-stim condition for patient 1. We have the stim condition for patient 1, right? Non-stim from 2, etc. So now any type of combinations that we want to make in t-SNE space or clustering space, will be very easy. So let's talk real quick about the plugins. We have Tisney built in, but let's assume that you guys want to do UMAP. Okay, how do I get UMAP into FlowJo? The easiest way to do this is to click the very top button here where it says FlowJo Exchange. Okay, this takes you to our website where we host all the different plugins for both FlowJo and SeatGeek, which is our single cell sequencing software. You can maybe turn that filter off since you're only looking for FlowJo related plugins. And then that will filter the list here for you. So you have a bunch of different tools. We're going to be using the Cluster Explorer here today in a bit. And then I was going to show you where you can find UMAP. So these are all alphabetically listed. So UMAP is going to be towards the bottom. Okay, we're going to see UMAP today, or at least we'll see Tisney. We'll probably see XShift, which is a nice clustering algorithm, as well as Phenograph, another good clustering algorithm, and maybe some others. We'll see what we have time for. So let's jump back here. Okay, so that's how you get the plugins. You download them, and when you download a plugin, let me just show you. Inside each of the, what will happen is they come in as a zipped file. So in this case, the other day, I downloaded the Sunburst plugin, right? And when you unzip it, it forms a folder, and then it will contain a series of files, the all-important jar file, as well as the how-to. So if you actually double-click on the how-to, right, this is a list of instructions on how to set it up. So some of these plugins require R, others do not. But you just follow the instructions, and then you'll be able to use the plugin for whatever purpose. Okay, if you have any questions, of course, we have our contact information there. If you have any trouble, please just send us an email or call us. We'd be more than happy to help you install those. All right, so that's how you get the plugins. Now, let's get back to our workflow. We got our categorical variables all separated. Now let's go ahead and do some dimension reduction. I usually lead with dimension reduction. I like to use Tisney, and I'll explain actually why I'm going to use Tisney here. Okay, let's take a look at this slide very quickly. Or maybe not that one. I want the one that has all four. This is a better explanation, or at least a better visual. So all the dimension reduction techniques, what they do, again, is they sort of project all of your data, right? All the different phenotypes of cells onto a single two-dimensional plot, which makes it very easy to like visualize all the data, right? It's right in front of you, all the different cells right in front of you. Cool. Now, each tool has a different way of displaying those data. UMAP and TriMap are said to preserve the global structure of the data. And what that means is that cells that are more highly related to one another generally tend to be juxtaposed closer to one another in this two-dimensional space than in Tisney. And the example I can give you here is, if we take, for example, the B cells, which are kind of outlined in purple here, or I should say kind of a pinkish color, right? And monocytes. These guys fairly closely related in terms of the lineage markers, et cetera. Whereas B cells and CD8 T cells or B cells and CD4 T cells are a little bit more divergent. So UMAP and TriMap tend to preserve those relationships, right, better than Tisney does. So in this case, you can see Tisney here has pink islands, right, for your B cells, and they're very close in space to your CD4s. Whereas in UMAP and TriMap, the CD4s and the B cells, right, are pretty far away from one another. Okay. In Tisney, all this means that is in Tisney, the white space between things is important, but where they are juxtaposed to one another is not important. Right? So it's not telling me here, because this pink island is very close to my CD4s or my CD8s, that they have some sort of lineage marker or cytokine marker in common. It just so happens that Tisney decided to put this thing there, and it did it. okay, kind of at random, so to speak. It doesn't necessarily, keep track of what all the other populations have in common, whereas UMAP and TriMap do. Now, TriMap, UMAP, super useful in the sense that, they have these little peninsulas or these projections that come off the primary bodies. That's really nice for trajectory type of analysis or developmental type of analysis, right? You've got some hematopoietic stem cells or, I don't know, some sort of lineage, early lineage cell, and you want to kind of track it. How does it change over time or as a result of my treatment? These little peninsulas kind of allude to the fact that you have these cells that are transition state cells, right? They're starting to produce more cytokine, or their lineage markers are moving, and they're moving in a particular direction, right? They're looking more like monocytes, or they're looking more like CD4 T cells, whatever the case may be. So really good, really powerful for that purpose. I prefer t-SNE only because, as you can kind of see in the differences between t-SNE and UMAP here, if I look at the B cell population, I can kind of infer that there are probably two different B cell populations if I look at the UMAP space, right? There's this little peninsula here, and then there's this globular cluster down below. My immediate assumption is, okay, one population here, maybe another population here. Now, if I look at t-SNE though, t-SNE tends to break things up on a more, like, I don't know, it's just more refined that way. And I prefer it for that purpose because it kind of reveals the nitty-gritty phenotypic details between populations, maybe sometimes too much. But when I look at this, I can see, right? I can see one island over here, another island over here, this island right here, right? And then this island off to the right that looks like it's kind of breaking off from the main one. So at the minimum, I could see maybe 3 different populations, and at max, I might be looking at 4 here when I just first glance at this. Okay, and same thing when you look at the NK cell population, which is in purple. Looks like there's at least 2 here, plus 1 over here, and another one over here. Whereas in, the UMAP, I don't know, you've got a couple of pops here, another one here, and then of course the globular cluster. Now, embed sum is kind of a mess. I don't like embed sum so much because the borders are very fuzzy. You don't know where one population begins and the other one ends. It's a little bit harder to see if there's multiple populations. They're just kind of all run together. So I don't really like that one for visualization. All right, so Anna asks, are there any plans to include non-random t-SNE initialization based on PCA or other global distance preserving method option in FlowJo soon? Yes, there is. So we have been talking about that, and I know they are going to implement it. The question is whether they're going to implement it in version 10 or version 11. So we're currently working on, building version 11, but V10 is still obviously being updated. We just had a release. So I caught wind of this discussion last week. And the thing is, it has not been settled as to whether it's going to be in V10 or if it's going to be in V11. I'm hoping it's going to be in V10 since we already kind of have the infrastructure there. So great question. And I just wish I had the, the immediate answer for you. Okay, so I'm going to close that one out and let's go back to this. So anyway, we have, again, these options. And as I said, I kind of prefer t-SNE as a, just a readout, but again, it's up to you what you wish to use. So we'll go ahead and run t-SNE on this particular sample, come into the parameter selector here, and we'll go ahead and deselect any of the parameters that are not important or that do not contribute any sort of enhancement to the signal to noise ratio, right? So for example, we don't need t-SNE to run over the viability die. We don't need t-SNE to use the dump channel, right? We've already gotten rid of these cells, and adding these parameters in is only going to contribute noise, right? If anything, it's just going to delay and sort of muddy the waters between the populations that we want to see in the end. So in any case, we're going to have our compensated parameters that are important to us minus the ones that don't contribute anything. Okay, now down below you have a bunch of different knobs that you could, that you can use to alter how Tisney produces the, the image. I highly recommend that you leave this on the default here where it says auto or opt-SNE. This was a modification that was developed by Anna Belkina at the Broad Institute. And it's a very good modification because it speeds Tisney up, gives you the result, in a much shorter amount of time than you would, have to wait sort of for it if it were the original implementation. So you could actually test this by kicking this over to the manual method and then running the, running the output. Do the same thing again with opt-SNE and see how quickly you come out with results. Okay, so down below you have some options here to choose the number of iterations. So Tisney is an iterative algorithm, right? It sort of looks at the landscape once, kind of tries to, see which populations of cells are present or if there's sufficient density. It goes through another round and, refines that landscape, so to speak, multiple times until the populations resolve themselves. Now, generally speaking, if you have, I don't know, 1 million cells or less, 1,000 iterations is probably a good rough start, good place to start. If you have more than that, you can bump this up to, I believe 3,500 is the maximum number of iterations you can give it. And then, of course, the perplexity. This is a value that is very similar to clustering algorithms' K value, like the K value for next nearest neighbors. It's similar, at least in that regard, but it influences. I find that it has the highest influence on the white space between islands. So, if you find that your Tisney isn't well separated or whatever, you can always come in here and play around with this number. I think 30 is a good starting spot, but it doesn't work for every data set. So just know that you may have to come in here. And my understanding of the learning rate is this is a knob that will either speed up or slow down the algorithm, but to some extent the opt-SNE kind of either usurps this or at least helps this so that this value is not so important in the outcome of the projection. Okay, so I'm going to leave everything here as a default, and then you have a couple of different further options down below. There's the KNN algorithm that you can choose. There's either the approximate or the vantage point tree. If you want to go super fast, right, your approximate is pretty good. This algorithm actually is used by Spotify to, predict which song maybe that you have not already heard, but might belong to your playlist. So it's a pretty cool algorithm. I think it works pretty well. My playlists are pretty decent, I would say. I'm not asking for anybody else's taste, but just saying. And then the gradient algorithm here, Barnes-Hutt, was the one that was published, I believe, in 2008 by Hinton. And then there was another update to that called the fast Fourier interpolation, which, it's a much quicker gradient algorithm. So if you wanted to go, I guess, as fast as possible with Tisney, these are the settings: auto, opt-SNE, approximate, FFT, and that will give you probably the fastest output possible. So let me stop here. If running manual, okay, so Anna's here. She could tell you all about Tisney. But she says, just use the learning rate as 1/12 the number of events. Okay, so 1/12 the number of events. I have 40,000 events. So 1/10 of that would be, 4,000 cells, or the value be 4,000. So I'm assuming 1/12 to be very close to that 2,800 value. Something a little bit less than 4,000, right? So hopefully this will be good. And then we'll go from there. But that's a very good tip. And thank you, Anna, for saying that. So just if running manual, just use 1/12 the number of events. All right, so we'll go here and we're going to click run. And then this should not take very long. Tisney will produce some results. In fact, I think that this version of Tisney is faster than UMAP. So if you actually try to do, one next to the other, Tisney will actually run faster. And I want to say historically, people were switching over to UMAP because Tisney initially was abysmally slow. And when UMAP came out, I think it was like a 10-fold reduction in the amount of time you had to wait. But I think opt-SNE definitely puts it on par with UMAP, if not, better. Then let me clear this out. Okay, so good time to ask questions if you have it. Obviously, we've got to wait for the results here. But it's approaching completion. And when it does, it'll usually pop up your t-SNE plot. Okay, so there's a question here. What is the maximum number of cells recommended to have in the concatenated file to run t-SNE? Well, I wouldn't, the RAM is probably the most limiting factor on most, computer systems. But internally, we've been able to run this t-SNE on the opt-SNE settings on 50 million events, and it will complete I want to say within 6 hours or 8 hours. I'm not 100% sure on the time, but we couldn't do seemingly more than that. And I'm not sure, that was on a machine that had 32 gigs of RAM. I'd have to check again and see what we've actually been able to push this to, if it's gone to more than that. So I'd say, just with a very cautionary statement here, I would say 50 million is probably your upper limit. But if you've got a nice beefed up computer, you might want to try more than that. But in general, 50 million kind of seems to be sort of the practical upper limit on most, desktop computers that aren't, fitted to be high performance, like ultra high performance type stuff. Okay, so we get our t-SNE plot here. Again, lots of different islands, et cetera, which is great. A lot of people would like to orient themselves initially on the map here to figure out, what expresses what. There's a couple of ways you can do this. You can click the color axis at the bottom left, right? And this will heat map now by a third marker. So right now we're looking at IL6 expression. You could go in here and maybe look at some of your lineage markers, for example, CD14, to have an idea of where your monocytes are in relation to everything else. Okay, that's great. I can see where they are, right? And then maybe I can go to the patrolling monocytes, et cetera. You could do that one by one if you'd like, or you can kind of do this all in one shot by opening up the layout editor and then creating the all famous figure that you see pretty much on every poster at any trade show. You come here, you right click on the t-SNE plot, and then you go down to the bottom where it says make multigraph overlays. And then you're going to choose the very bottom option there in the side menu that says multigraph color mapping. Okay, this will give you a little prompt. It asks you, okay, well, what do you want to heat map, right? Or what do you want to color map? So I'm going to go ahead for all the parameters, again, minus the viability die. Probably don't need the dump channel. It'll take a second or two, but what ends up happening here is it produces, that figure that I was talking about at all the trade shows for every poster, right? Everybody has this, so you kind of can orient yourself, right, on the t-SNE map, so you can see where the different types of cells are, where the different expressors are, et cetera. Right? So it's a very nice, convenient way to sort of visualize the data right off the bat. Now, this is great, but it has nothing to do with our categorical parameters comparisons, right? We really want to be able to compare experimental variables. So the way we do this, I'm going to create a new workspace here, or a new layout. And really what I want to do is maybe compare the non-stimulated condition, right, to the stimulated condition. Let's start there. So we drag these into the layout editor, we overlay them. I'm going to get rid of the page breaks here real quick. Okay, and then we just want to convert the graph here, right, over to t-SNE 1, t-SNE 2, right? Something like this, so we can see the overlay. Now I would go one step further, and I would recommend, rather than looking at this as a dot plot, I would actually prefer to look at this as a contour and maybe show the outliers here. Okay, the contours are kind of cool because you can see areas of overlap a little bit better than you can with the dot plot. A lot of times when a dot plot is used, the sample that has more cells in a particular area will obscure the one that has fewer cells. And even though, you know, there may be overlap there, you may not always see it. But with contours, it's always present, so you can see it. So I'm going to zoom in here as much as I can so that you guys can really see this. Now, how cool is this, right? All we've done is taken our two categorical parameters, right? The stem and the non-stem, and we've just overlaid them here in t-SNE space. Right? So the red sample here, right, represents the non-stimulated condition, and the blue sample represents the stem. Right? You can see that there are areas where the blue sort of stands alone, right, all by itself. Right? And then there, conversely, I guess, there are areas where the blue has a lot of overlap with the red. Okay, in the areas where the blue is by itself, right, these are phenotypes of cells that are unique to the stem condition, right? They only exist in the stem condition and not in the unstimulated. Okay, vice versa, right? There are areas that are all red here. These are, you know, baseline state cells, probably not expressing much in the way of cytokine, right, that occupy only, you know, this portion, or these portions, I should say, of the t-SNE space. Okay, in the middle, there are areas here where you can see there's overlap between the stimulated and the non-stimulated. Okay, and obviously the frequencies are different, but the distributions or the phenotypes may be the same. Right? It's just there are more, let's say, of the blue or the stimulated cells here in this middle set of islands versus the non-stimulated. So number one, right, very powerful visualization that way. And then you could say, all right, well, I see that there's differences between these two categories. How do I identify these phenotypes, right? How would I go about that? Well, there's a couple ways you can do that as well. And this is the way I would recommend. Let's assume for a second that you're interested in the stimulated populations, right? You're interested in the stimulated phenotypes. So what you would do is hop back to your stem population here, open that up, okay? Make it look like what you have in the layout editor. So we're going to change this. Instead of being a histogram, let's change it over to t-SNE1, t-SNE2. And then I would go a step further and make this, I'll discuss this a little bit further, but let's just put it as a contour plot to begin with. Okay, and you can see, all right, this is the entirety of the stem. I can kind of see, oh, this island down here is probably, that's all blue or mostly blue. Maybe I want to, you know, isolate this population and investigate it a little bit further. You can go ahead and create a gate. I would recommend too, though, that you also take a look at this kind of once you orient yourself with the plot that you have in the layout editor. You may want to look at this as a density plot. And the reason I recommend the density plot is because the contours, even though they're good at sort of showing you where the, you know, by looking at the topology, you kind of have an idea of where different populations exist, right? Different phenotypes. But the density plot really kind of gets down to the nitty gritty, right? You can kind of see, for example, in this little blob, there's a pocket of cells over here, as well as a pocket of cells over here that are likely distinct, right? Because of a marker or two, whatever. Okay, maybe not as obvious here in this population of cells that you have kind of elongated in this section, but who knows, maybe this population over here that makes the foot is different than the one at the top of the head. In some of the other bodies over here, you can clearly see that there are dark patches kind of separated by white space or a little bit less dense areas. Right? And these can be, again, those little nitty gritty populations that might be more or less different from one another. So a lot of times I'll come in here and I'll just go ahead and just, you know, create a gate on an area that I'm interested in. And I usually refer to this as my query, but you can call this, I don't know, whatever population you want, pop1, pop2, blah, blah, blah. So you create your query and then you can come over here and you can dump it on top of your overlay. And then from here, what I recommend is just change the color to something dark and contrasting so that you can easily see it on the white background. Sometimes I even fade the colors, you know, of the stuff I'm not really interested in into gray or, you know, lighter colors. But in any case, once you have the overlay created, what you now want to do is you want to see the phenotype, at least qualitatively, right, of that population that you've isolated. So you'll right click, scroll to the bottom here where it says make multigraph overlays. And then I recommend that you choose histograms here. If you're a glutton for punishment, maybe you want to do N by N, but I would recommend histos and then the histograms here. Right, we'd have to stretch this out a little bit, but in any case, this gives you a panel with a line trace of all the different antigens that you have present in the data set, along with some other parameters as well. So I recommend too that you trim this list. So right click on it after you create it, scroll down to the bottom where it says select multiple. This just makes it easier to quickly select the parameters that you're really interested in and dump the ones that you are not interested in, right? And that will trim the list for us. And then we can come here and take a look at what the phenotype of this particular population happens to be, right? Again, from sort of a qualitative standpoint. So whatever these guys are, IL-6 negative, they're DR positive, which kind of makes sense. We really isolated DR positive cells to begin with. Okay, probably mostly negative with respect to CD1C, so not a ton of dendritic cells here, although there are some here that seem to express a little bit, but you can kind of see, right? This little tail here, these are probably the true positives, and these two distributions here, right, correspond to this sort of bubble population that you see. And let's go back to this graph real quick. And if we look here, you see how there's kind of these two lobes. Those are going to be these two distributions. This little guy over here, what they were considering positive, right, is this stuff over here. So I would say that these are probably cells that fall into the CD1C negative category as judged by our manual gate. And we can actually overlay that in a second to see exactly whether or not they are placed there. Okay, so it's negative for TNF, activated though, 83 is present. It's not monocytic in the sense that it doesn't express CD14, no IL-1. Right? What is this guy? Okay. And it's CD123 expressing, it's a plasmacytoid dendritic cell. So I'm not surprised by the sort of lack of pro-inflammatory cytokines there. Okay, so we're locking onto the PDCs over here. Now, if you want, and just, you know, to sort of confirm your findings that this is not a, you know, CD1C expressor, you could take either your CD1C positive gate, right? You could take that and then just drag and drop it on top of the t-SNE space and just see where that maps to. Okay, you can obviously see here in the histogram, yes, that little section I was telling you about is kind of that tail end of that population. Okay, but again, now we have to kind of look at this with, you know, some different, different colors, and maybe it would help if I convert this back to a dot plot. There's very few cells in this population, and so we're going to have to probably switch around those pops to see exactly where they land. But let's see what happens here. Can I find these guys? I don't see the green. And I probably should pick another color, right? Because there might be people that are a little bit colorblind that way. So let's choose pink. Interesting. So it updated my line trace, but I don't think it's updated my, t-SNE plot. Oh, I know why, because I'm pulling this from another sample. So it shouldn't do that. Anyway, my bad, my mistake. I need to really transfer these gates onto the concatenated file to see where they map to. So that's my mistake. Well, let's go ahead and drag this right onto the concatenated file. And now let's go ahead and let's get rid of this guy real quick. Let's take that from here. I get sidetracked very easily, so I apologize for that. But here we go. Don't. The Boeing, the horrible Boeing sound. But there we go. You can kind of see, okay. Right, the orange dots corresponding to our CD1C positives, right? They don't map to that area where we had our query gate, right? Which is this one that's kind of in that dark blue, the dark purple. Okay, so that kind of makes sense. Now, if you wanted to, there's a couple of things you can do here. You can, right, explore other phenotypes that are in this t-SNE space if you would like. So if I go back to the stem here, right, I could create other query gates and just simply drop them on top of the plot we have here. Or, right, I can roll this guy around. So I can go looking at other populations in other sections of the t-SNE plot. Right? And when I do that, the line trace will update. So if I come here, let's go ahead and put the query on the top again so that we can actually see where that, just where that position is kind of in the forefront of things. Right? In this case, we're definitely capturing some of those IL-6 producing dendritic cells. So these guys are a little bit more on the pro-inflammatory side. Right? We got some that are primarily TNF expressors. They also express IL-1. Right? There's a bit of IL-12 as well. Negative for 123 and IL-10. So it's kind of a qualitative assessment, right? Yes. So, okay. Thank you, Anna. Okay. So, in any case, you can roll this around to look at the different phenotypes. But the other thing that you can do here, if you want to be a little bit more quantitative about it, is you can always put these statistics underneath your query gate, right? So you can come here. I recommend using this tool. So if you go up to the workspace tab and you go here to the statistics band, you go to the median, right? The little median drop down. At the very bottom, you have this very handy little option that says all compensated parameters. And that will show you, right, the MFIs basically for each of the markers. And of course, you can export this in a form of table if you want to use it in Excel. Okay, and just keep in mind though, if you're rolling this gate around, these stats are going to update accordingly, right? So move this around, so on and so forth. You can see how the values update. All right, so that's how you would do categorical sort of, comparisons in t-SNE space. We could do this with the patients, for example. So for example, if I come here and create a new workspace, I could compare, let's say the stim condition. We're really interested, I don't know, in the stim condition of patient 1 and patient 2. Right? So I just take those two categorical gates and I overlay them on top of one another. And I come here and I say, okay, well, let's again look at this as a contour. And obviously we want to look at this along the t-SNE axes. Okay, so we go ahead and display that. Now in this case here, if I zoom in a little bit, so you guys can see this a little bit better. In this case, if we assume that both of these patients are healthy, then we would expect that they're going to have the same distribution or at least the same types of cells, right? Maybe the quantities are different, but by and large, we have overlap here, indicating that these two patients share the same phenotypes of cells. We don't see anything, out of the ordinary. Okay. You could do the same thing though with the non-stim condition, right? Just say, okay, well, how do their baselines compare? You overlay them, come over here, right? Change this over to a contour plot and do the same thing. t-SNE 1, t-SNE 2. Right? So again, if they're healthy, I should expect that, these guys are going to have similar distributions, maybe different frequencies. Right? But regardless, we can compare the two in that same space. Okay, now if I had a more complicated example, I think you guys get the idea. You can just mix and match your categorical gates, right? And then look at it in dimension reduced space, whatever you want to do. Okay, so that is dimension reduction. Let's take a look at clustering. Okay, so I'm going to go here to the concatenated file again, and we'll go up here to our workspace tab. We go to the plugins drop-down menu, and from here we can choose the clustering algorithm of choice. So there's several different clustering algorithms that we have. There's XShift, there's Phenograph, and then there is FlowSum, as well as FlowMeans as clustering algorithms. And I will say that, I don't know, to me, like in most papers that I read about clustering algorithms, when they use them, Phenograph seems to have the highest F measure that I've seen. And so the F measure is a score that is given to algorithms to see how well they fit the manual gating standard. So Phenograph does a pretty good job of mimicking, or at least it gets as close to, you know, the human gates, I guess, as possible. Other algorithms like FlowSum are also good. They're just not quite as high. And I mean, this is like splitting hairs. You know, it's not like you would only use Phenograph. You certainly can employ any one of these three, I think, with confidence. FlowMeans, on the other hand, even though it's very fast, it doesn't do a great job of cutting up elliptical populations as well as the other three do. So this looks for really round populations. The other ones seem to do a better job when you have populations that might be smeared. So we'll come here, let's choose Phenograph for our first run. And what we're going to do in this case is, again, select the compensated parameters, dump the liability die and the dump channel. Now you have a choice here with the K value. So the K value, it's basically asking you how many points, data points, does this have to count in its initial initialization, right? So the algorithm essentially starts itself randomly somewhere in the data set. But you can kind of think of this number as the minimum size of a population. So if you're looking for rare cells, the K value technically should not exceed the number of rare cells that you expect to find in that data set. So if you have, let's say you expect to find 15 cells in in your given data set for that rare population, then your K value, if it's 15 or lower, it gives a higher probability that the algorithm will actually capture that rare cell population. If the K value is higher, what ends up happening most of the time is that it will incorporate that rare cell population into a larger population. Okay, now that's not always the case. It depends, of course, on how well that rare population of cells separates itself from, the rest of the mix. So if those rare cells are a fuzzy CD1C positive population, the odds are, Phenograph and other algorithms are not going to be able to capture it as a nice pure population. But if it is well segregated, like a CD4 or CD8 population, double positive for a couple of different markers that no other cells express, it should be pretty easy to pull out. But in any case, the lower this value is, the more clusters you're going to get out in the end. A lot more backgating or figuring out which cells belong to what population. The higher this value is, the fewer the number of clusters. So it's basically an inverse proportion. Now, why would you ever use a clustering tool in the first place, since it doesn't give us a visual display of any kind? Well, one of the things it's good for is it calculates or analyzes the data, crunches through it fairly quickly, versus manual gates. It's very fast. The other advantage, though, that I think that it gives is that these algorithms can give you a fresh perspective on the data. A lot of times they can pull out cells that are like these transition state cells, again, mid-expressors for either lineage markers or cytokines that a lot of times we may not necessarily be privy to, or at least we're not really thinking about in our initial analyses, right? A lot of times I go through my gating and I'm like, this is positive, this is negative. Don't really care if there's something in the middle, or if there is something significant in the middle, maybe I'll gate on it, type of a thing, right? But if I had to do that for every 2x2 combination, I'd probably pull my hair out, right? So these algorithms are really nice at pulling out some of those mid-state populations, and at least, you know, gives you something to look at. Who knows? Maybe there's something there, a phenotype that is driving the differences that you see in the data set. Now, I realize that I probably should have explained that while it was running, because it will take a second or two here to run. But regardless, that is some of the rationale that I would, you know, that I would use to, you know, to run this type of a tool, or these types of tools in your analyses. So this is a good spot. If you guys have any questions, go ahead and ask them. And maybe what we'll do here is we'll run a couple of different algorithms and see how well they, you know, cluster the data, or how do they compare to one another. The primary thing I want to get to is, again, how can we compare categorical results or categorical variables in cluster space, right? So give me a second here. Phenograph finished. Let's go ahead and just follow this up. We've got 16 clusters from that K value of 30. So let's just try the same thing with XShift, right? Come in here. XShift is a clustering algorithm as well, works a little bit differently than Phenograph does, but we can set a lot of the same parameters here. So number of next nearest neighbors, we can say that K value, let's make it the same as what we had for Phenograph, which is 30. The distance metric here, Nikolai Samusic, in the paper that he wrote on this, he recommends that if you have 10 or more colors, you should be using angular distance rather than Euclidean. So you actually have the choice here of which one to use. So we'll leave it at angular. You can actually subsample within this algorithm as well. Obviously, we have 40,000 events, so we're not really subsampling at all, but you could reduce this if you want it to run faster. But I assume that you guys will have downsampled to begin with anyway, but it's kind of a nice little addition here. Okay, so we'll go ahead and click run, and then just see what, you know, how many clusters does XShift come up with. And then there are some things that we can do in the Cluster Explorer to kind of compare the results that we get here. But the primary one that I want to get to is just to show you how you can compare categories here with respect to the each of the clusters. Okay, so XShift is usually pretty fast, and let's collapse these gates so we can see them. Okay, so in the case of XShift, much larger number of clusters here. We've gotten 40 versus the 16 that we got with Phenograph. Okay, so this is not exactly an apples to apples comparison, but regardless, we'll see what happens here. Okay, let's just focus on categorical kind of comparisons. So in order to really efficiently look at the phenotypes of these clusters, I would recommend that you use the Cluster Explorer. Okay, you could do this in the context of t-SNE space, right? We could do this like so. We drag in our t-SNE, and then we take our, you know, Phenograph clusters. We come in here, right? Dump them on top. Turn this into a t-SNE plot. And then kind of do the same thing that we had done with our query gate, right? Actually, let's move this off towards the side here. And you could do something like this where you say, okay, show me those histograms again. Now, this gets really hairy, right? I mean, trying to look at all these clusters, you know, picking apart one versus the other and seeing what they express. This is not very efficient, and I wouldn't recommend that you do that. But just to show you that you could if you wanted to, and in fact, that was kind of the old school method of doing it. But now we have the Cluster Explorer. So here we go. We're going to say Cluster Explorer from the plugin menu, and then in order to use this tool, okay, the important thing is you have to have run at least one clustering algorithm, okay, and some form of dimension reduction. So t-SNE or UMAP, otherwise it will not work. If you just do one and not the other, you can't use this tool. So it sort of forces you to use the high dimensional tools. Okay, dump what you don't want to look at, click OK. And then this is going to build a few windows for us. Okay. On the left-hand side, it's going to have the relative numbers of events versus all your cluster numbers. So you know which ones are more populous than others. In the middle, you have a line trace graph, which I think is an extremely powerful and useful tool. You can interpret the phenotype of the different clusters. I apologize, I clicked off of here because the zoom bar is blocking the top of this. I want to bring it down. So I clicked on this line trace and it went gray. So I'm gonna have to be careful about how I dance around the zoom bar. But in any case, you have the line trace here that kind of tells you, right, who the IL6 high expressors are, DR, all the other parameters that you had selected in that parameter selector, right? On top of that, not only does it give you this nice visual display, but it also allows you to gate on it. So remember how I told you that these algorithms tend to find populations that are sort of mid-expressors for different cytokines or lineage markers? You can kind of see here, right? Phenograph has kind of pulled out two TNF populations that seem to be in the mid-level of expression. If you really want to zoom in on those, study those in a little bit more detail, you can gate on them here, right? So that will go ahead and isolate those two clusters. It'll sort of back highlight them onto the heat map table that you have down below. And of course, in the number plot that you have over here, it'll actually highlight the cluster numbers that you are that you're looking at. And of course, you can move this gate around, right, one place to the other, and you can combine it with Boolean logic, right? So you could say, okay, I want TNF, but I don't want things that are non-monocytic, right? So you could say, all right, exclude all the negative monocytes, whatever, right? So you can use sequential Boolean logic here to help you, okay? And then just click the clear gates option here to get it to go back. Now, down below is the heat map table, and this is the sneaker surprise. It seems like it's a boring heat map that kind of shows you, okay, here's cluster number 1, 2, blah, 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 and all of the phenotypes, right? But this is the tool that you would use to compare your categories, right? So a lot of times when you run these clustering algorithms, as I mentioned, you don't really know what the phenotypes are until you get here. But the other thing that you would want to know about these clusters is how do they skew relative to my categories, right? How do these clusters skew in the stim versus a non-stim? Are these all sort of evenly distributed across the stimulation and non-stimulation conditions? Probably not. But I don't know offhand how they do, how they skew unless I use this tool or do some fancy footwork in the workspace. Okay, so from here, what I would do is click on the option drop-down menu, and what you want to choose is this option that says add nodes as columns. Okay, and then that brings up a little window that looks like your gating hierarchy. And then from here, you can say, okay, well, I want to look at the non-stimulated condition, and I want to compare that, let's say, to the stim. Right, so you can say select new columns, and then that will add the two categories. In my case, it could be multiple categories in yours. But the bottom line is now I have a column for the non-stimulated condition versus the stim. Right, so now I'm just previewing the experimental variable here, stim versus non-stim, and looking to see how the clusters are associated, right? Which clusters are associated with which condition? Well, according to the heat map, the yellow color means that there are more cells, right, in that column versus the blue. So in the first handful of clusters that I have here, right, it looks like clusters 1 through 5. You can clearly see that these are bright yellow, right? The non-stim condition is bright yellow. And this means, right, that these clusters 1 through 5 are actually identifying cells in the baseline state or the non-stimulated state. And if you actually take your mouse and kind of hover over one of the cells, you can see how many events are in it and the frequency. Okay, if I move next door to the stim over here, you can see that cluster number 1 has very few events that come from the stimulated condition. So, you know, the stimulated category, only 40 cells, right? Roughly 1% of total. So the first 5 clusters are probably identifying basal state cells, not activated or anything like that, okay? But cluster number 6, right, all the way down here through cluster 14, and some that are a little in, you know, I guess 16 here, right, 16 in particular, definitely on the pro-inflammatory side, right? It's quite the opposite. Cluster number 16, if that's the choice, you know, you're really interested in what this guy happens to be, you can always come over here, just click on cluster number 16. It'll isolate the line trace for you, and you might get a better idea of its expression, like what is the phenotype here, right? Obviously, MHC class 2 positive, right? Probably a negative, you know, for CD1C is my guess. It's that odd negative distribution that's kind of wide, I think, that this is going to fall into. Mid TNF expressor, right? Kind of mid with respect to CD83. And then, of course, we seem to be low on a lot of other markers as well. So I'm not exactly sure what this phenotype is, right? But it does have some pro-inflammatory characteristics. Okay, clear this again. Let's clear the gates. Right, to preview that heat map again. And then you can pick out, you know, any other cluster that you're interested in. You can highlight it here as well. You don't have to go to the number selector if you want. Okay, but that's one way that you can visualize the categories, right, and make comparisons between those categories with respect to the skewing of the clusters. I really like this table for that purpose. Now, for the other menu items or the other windows that are sort of popping up in the background, you have the t-SNE plot here by default. So again, if you select a given cluster, you can see where those cells map to on the t-SNE plot. You don't have to look at it in the context of t-SNE. You can create another 2-dimensional plot here to have in the forefront, right? Maybe you want to look at IL-6 expression. You could create another one that maybe looks at, let's say, you know, that one cluster that we were looking at before, we were sort of skeptical about its CD1C distribution, right? So if we go here and now we say, let's look at cluster 16 again, right? With respect to CD1C expression, right, where I'm looking at it here, I can clearly see that it is in the negative end of the graph, right? We've got this really wide spread negative population. The positive stuff is most likely what we had out here that we kind of were pressing up against the axis. Okay, so I probably could have done a better job of transforming this. Maybe narrowing the negative population would have done a better job than maybe stretching it out as much as I did. Okay, but in any case, by looking at this graph, I can clearly, I'm confident, right, that this is not a dendritic cell, although it does have some pro-inflammatory characteristics. Okay, negative with respect to IL-6, et cetera. But you can put as many of those plots as you want. Other things to say on this menu, you can switch between cluster sets. So we're looking at phenograph right now, but we could switch this over to XShift. Obviously, XShift had a heck of a lot more clusters. So again, if you wanted to look at some of these mid expressors, right, which we have a number of, this is one thing, you always have to remember to switch this back to and. Maybe you want to go for, you know, TNF rather than CD1C, but in any case, right, go ahead and highlight those. Back highlights everything back here. Okay. Last thing that I'll mention on this particular cluster explorer is you have the ability to sort of mathematically compare results between XShift and phenograph. So sort of qualitatively, you can see where each of the clusters kind of map to on the t-SNE space. But you can also go to this button where it says compare to, and then you can say, okay, currently we're looking at XShift. We want to compare those results to the phenotype results. Click OK. And then what it's going to do is it's going to put the XShift clusters, in this case, on the x-axis, or sorry, the y-axis over here. And then the phenograph clusters are on the y. And then the tool here just tries to arrange, you know, cluster for cluster what they are. So phenograph cluster number 14 and XShift cluster number 14, in a mad coincidence, are actually probably the same population or mapping to the same population of cells, okay? Right now, the graph is set to look at mutual events. So whichever clusters sort of share the, the highest numbers of events are obviously going to be matched up that way. Cluster number 9 of phenograph and cluster number 26 of phenograph are most likely the same with, very heavy overlap. You can kind of look at that in comparison to all the other combinations that are present for 9 and whatever other cluster number from XShift, so on and so forth down the list. Now you can modify this to look at the Jacquard index or the F measure. So the F measure is kind of a more complicated measure. This is known as the harmonic mean, or the ratio between precision and recall. So I should have brought up a slide that kind of shows the mathematics, but it's kind of, in its simplest sense, basically what we're looking at is we're looking at the quantity of false positives, true positives, right, and true negatives, kind of all together. So you have matching events, you have matching excluded events that the F measure sort of takes into account. And in fact, the F measure, again, is usually what is published in a lot of papers when they compare an algorithm's performance to manual gating. But you can also use the F measure to score two algorithms, in this case, to one another. Okay, so in this case, a perfect value would be 1. And in this case, the closest we get to 1 is about 0.85 between the two clusters, or the two clustering methods. Okay, the Jacquard index is a ratio of the one algorithm's results versus the other algorithm's results over the sort of the union between them. It's kind of hard to explain verbally, so again, I apologize, but you can definitely look this up. Even Wikipedia has a great definition, great visual for what the Jacquard index does. But in any case, these are just different methods to compare mathematically the outcomes of the different algorithms, if you wanted to sort of show that. Okay, now all of this stuff can be copied, pasted to your downstream program of choice, right? You can copy any of this stuff. You can export the table information, blah, blah, blah. It's just a right click, okay? And then you can get there, get to Excel or get to Prism, etc. All right, so for me, that kind of concludes the areas where you can compare these categorical parameters, okay? I highly encourage you to use those clustering tools to kind of get to this stage where you can kind of see these interesting populations. And who knows, if you see them consistently pop up in your data analysis, it may be worth chasing down in the form of sorting, or perhaps, looking at these cells in historic data to see if they crop up there as well. Who knows, maybe you have a novel population or a transition state cell that absolutely must exist in order for some downstream outcome to be present. Okay, with that, I will say I'll take any questions that that you guys have. I'll wait around a few more minutes. And if I don't see any questions, I'll bid you all a good rest of your day. And I hope you have a good weekend. I really appreciate you guys attending today's session, and I hope you found it helpful. It's very easy, I think, once you create those categorical parameters. I think it's just absolutely necessary when you want to do this stuff. Okay, so there's a question here about the access to the recording. Yes, so let me show you where it's going to be posted. How's that? So we go here to FlowJo.com, any of our FlowJo websites, they all have this tab at the top. So I'm actually on the Exchange site, but if you went to FlowJo.com, you'd see the same thing. And you want to click on the Learn tab over here, and you will see that we have this webinars option, right? So it's learn webinars. And then this is probably where you guys were today when you clicked on it, or perhaps you got it in an email or something like that. So I'm talking about the high dimensional tools. Great. But at the bottom of this list, right, is our recorded webinars. So you click on this, and it will be posted here probably sometime later today. It'll be at the top of the list because it's the most recent one. So go ahead and check that out. So that's how you will have access to the recording. And I believe you can download like an MP4 file or something similar. Okay. All right, so that answers that one. Let's see what else we have in the chat box here. Is there a way to label or name each cluster? That is, yeah, cluster 1, cluster 2, sure. Unfortunately, right now it is manual. Okay, so the question is, can we rename these, right? So let's say, let's look for something here let's look at a phenograph cluster since there's fewer of them, and that way it'll make it easier. We go here to phenograph and we say, all right, these we know are to be monocytes, right? So let's just pick, I don't know, this guy right here. It's a TNF expressing monocyte, right? So this one happens to be cluster number 10. I go back to the workspace and I can say, okay, rename. So you right click on it and you could say, CD14 positive, TNF positive monocyte. I don't know, right? So you can rename it whatever you want. We're working on a plugin to sort of auto classify these guys. It's just, I feel like it's sort of still in its rudimentary phases. It's not super useful yet. I think it's even available, but I don't recommend it. I don't use it because I just don't think it's on top of its game. Let's just put it that way. All right. So any other questions? It's about 11:41 on my end. And if you guys have any questions, I'll hang out for a few more minutes looking at either the Q&A box or the chat box. And if you happen to have questions, I'd be more than happy to answer them. If not, again, have a great weekend. Have a great rest of your day. And use these tools. They're there to help you. I think they're, at the very least, they're kind of fun, right? They're just sort of fun to kind of explore the data, kick it around. I'm actually looking at a data set right now that's, you know, just kind of wild. It's all over the place, but it's really just kind of neat to kind of poke around and see what is and is not present. And I'm going to sip my coffee, so sorry for the slurping. All right, maybe wait another minute here to see if you guys have any questions. And if not, I will go ahead and close out today's session. All right, I don't see any other questions. So with that, I'm going to go ahead and close it out. Thank you all for attending today's session. Take care.